Welcome everybody, dear friends, colleagues, family members, dear candidate, parents, member of the reading committee, Corona members, and also the supervisors. I hereby open this academic ceremony in which Yuewei Cao will defend the academic thesis entitled I See, I Hear, I Learn. Audiovisual processing differences in dyslexic readers. And I kindly invite you now to give us a summary of your work. Thanks a lot.
Thanks. Members of the Defense Committee, family, friends, and colleagues, today I will present my PhD work on audiovisual processing differences in dyslexic readers. So let me start with a little background. Reading is an important skill that most of us learn early in our lives. When we read, we need to connect what we see, symbols and letters, with what we hear, speech sounds. For most of people, that happens automatically. However, dyslexia is a common learning disorder. People with dyslexia have normal intelligence, but struggle with reading accurately and fluently. For people with dyslexia, this later sound connection doesn't work so smoothly. So here is a fundamental question. What makes learning this connection difficult for people with dyslexia? So in my PhD work, I investigated how do adults with and without dyslexia learn new symbol speech sound associations and integrate audiovisual information. Regarding the first question, I investigate learning trajectory and whether similar sounds processed differently than dissimilar sounds. Regarding the second question, I compare two kinds of visual cues, written text versus lip movements. So to address all these research questions, I conducted 3 complementary studies. For Chapter 2 and Chapter 3, I investigated learning dynamics and phonological similarity. In Chapter 2, I used EEG to track the real-time brain activity. And in Chapter 3, I used a behavioral test to testing learning under increased task difficulties. Then in Chapter 4, I focus on audiovisual integration. So in Chapter 4, I compare text and lip read different visual cues, and I use fMRI to measure the underlying brain activation. Let me walk you through each study. So the first study, Chapter 2, I use an artificial symbol speech sound learning task. During this task, participants will see an artificial symbol, like unfamiliar letters, and at the same time, they will hear a familiar Dutch syllable, a Dutch sound, for example, ba. So their task would be to judge whether they are matched pairs or not by pressing the button. After each response, they will receive feedback. A happy face to indicate they are correct, and an unhappy face means they are wrong. So gradually, participants will learn which symbols goes with which sound. So they will have 6 pairs to learn within 4 blocks. And we have 2 kinds of sounds, similar sounds, ba, da, pa, and dissimilar sounds, fa, ka, la. During the learning, we recorded the brain activation using EEG. So the previous study, we could get the learning trajectories, and then we would like to investigate learning outcomes. So we use kind of behavior post-test. In this task, we would like to know how well participants could apply these new symbols. So we ask them to reading in the new symbols. So these are some scripts, but written by the novel symbols, novel artificial symbols they have just learned, including syllables, individual letters, and words. Okay, so let's see the learning trajectories. The first part is the accuracy trajectories. As you can see from the plot at the beginning, typical readers and dyslexic readers, they are all at the chance level around 0.5, and across the learning, they could achieve a higher level of accuracy. And generally speaking, they have quite comparable performance, but similar pairs are more hard to learn than dissimilar pairs with a lower accuracy. And for response time, we could find that these also have quite comparable performance, and similar pairs are also more hard to learn than dissimilar pairs with longer response time. But when speaking about the post-test, that task we asked them to read novel symbols, we could find readers with dyslexia show lower performance, which means even for the initial learning, they did a really great job. But when applying these associations, they will find it quite challenging. And for the EEG trajectories, I'd like to show this very interesting plot of one of my electrode clusters. As you can see in window S4, at that time window, there's a brain activation reduction, which shows our brain could process more efficiently for the established pairs. And phonological similarity or sound similarity didn't affect the learning process so much. And also between these two groups, we didn't find so much differences, only for some certain time window and for some certain electrodes we found subtle differences. And then in chapter 3, we use a quite similar task, but we increase the task difficulties. We added more pairs from 6 to 8, and participants need to learn within 6 blocks. And we also have 2 groups of sounds, similar sounds, ba, da, pa, ta, and dissimilar sound, fa, ka, la, ma. So the task is quite similar with EEG study, but during our behavioral study, we found very interesting outcomes. For example, similar to the EEG study, we found that participants found similar pairs are harder to learn, and also we found both groups have quite comparable performance, but there are some new insights came from this study I would like to show to you is our regression analysis. So when we do the regression analysis, we first do the curve fitting, and we drive in 2 parameters from the accuracy curve. One is initial learning level, another is learning speed. So we found only under the similar condition, these 2 parameters could significantly predict reading in symbols performance. So let me explain in that way, which means only when participants learn about the similar pairs, their learning performance could predict their learning outcomes of reading the artificial symbols. So for me, that would be a really interesting pattern. And next, I would like to show you my fMRI study. So first of all, I would like to explain what is audiovisual recalibration effect, which means if you are presented with an ambiguous sound, and at the same time you will see some very clear visual information, our brain will shift and we will hear that sound more differently and more close to the visual cues we have seen. And that is how in my task works. So we will present a very ambiguous sound to the participants, which means for half of time, they will think this is a bar. And for another half of time, they will think this is a da. So actually it's 50-50. And next, we will expose the clear text with a bar or a video of someone saying a bar. And we will measure whether participants will shift their responses to that clear a bar, and they will more likely think this ambiguous sound is a bar. And the same procedure for a da sound. So let's check about our results. So behaviorally, we did this task both in our fMRI scanner and offline measurement, and for both condition and for both lip condition and text condition, we all find quite robust recalibration effects. And we found that effect actually also quite comparable between typical readers and readers with dyslexia. Also, lip condition showed a much more stronger recalibration effect. And for fMRI results, I'd like to show you this plot. And from the orange part and the red part, there are some brain regions involved that's most important areas for audiovisual integration. And for our whole brain fMRI analysis, actually for typical readers and readers with dyslexia, the pattern is quite similar. So they all activate this important area. For example, the bilateral fusiform and the bilateral STG, STC, parts like this. And when we investigate the group differences, we could find that for bilateral fusiform, this is quite important area for example, visual information processing. We could find that typical readers have a lower activation than readers with dyslexia, but only under the lip read condition, not in the text condition. And another interesting finding is that we did a correlation analysis between the left fusiform activation and the phonological skill of the participants. We found only for that readers with dyslexia, when they have poor worse phonological skill, they will have a higher brain activation at left fusiform, which means they rely more on the visual information when they process the lip movements. And to conclude my key finding, so first of all, we find both groups could learn successful with similar neural patterns, but dyslexic readers, they show poor symbol reading performance, which means maybe for readers with dyslexia, they are not suffering from the learning deficit, but they might have some difficulties or challenges in automatization. And the second part is even under increased complexity, their learning trajectory remained quite comparable. So sound similarity actually affects robust across both groups and both studies. So we could say maybe sound similarity actually is a universal learning constraint. And third, we find comparable behavioral recalibration effect, but we find different neural activation patterns, which means maybe for dyslexic readers, they could use alternative pathways to achieve the same behavioral goals. And in my study, according to my finding, maybe for the next step, the intervention or early prediction, we could use this information to extend the practice on automatization, and maybe this would help people with dyslexia at their early age. And at last, I would like to thank you, my supervisor, Milena and Francisco, and I would like to thank you all the participants who support my project make this project possible. I would like to thank you, everyone who supported me. Thank you so much for your attention.
Thanks a lot, dear candidate, for this very nice, clear and also timely presentation. We open now the opposition, and I kindly ask Professor Dr. Sonja Kotz to, as the chair of the reading committee, to get a start. Professor Kotz is expert in neuropsychology and translational cognitive neuroscience at this university. Sonja.
Dear candidate, first of all, thanks very much for this very nice summary of your results. It was a great pleasure reading your work. It's a very coherent series of studies with some puzzling and interesting results, but very nice to understand this complexity of the phenomenon on dyslexia. I will ask you some specific questions about Chapter 2. And I will go one step at a time with my questions. So I'm very curious in terms of the behavioral data where you show very clear learning in both groups. So accuracy increases, reaction time decreases, and robust phonological similarity effects. You modeled that with particular power functions to capture the individual learning parameters. But in the EEG or the ERPs, you reveal early mid-latency differences. So there's a dissociation between the behavioral results and the ERP results. How do you explain these differences between the two measures?
Okay. Highly esteemed opponent, thank you so much for your kind compliment, and thank you so much for your interesting questions. So first of all, I would like to repeat your question to make sure I understand correctly. So you asked me, I found quite robust, for example, group differences. I found quite robust behavioral differences during the behavioral part, but for EEG, seems like the pattern is not so clear. Is that what you mean?
I see differences between the behavioral results and the ERP results, and I'm curious to hear from you why you think these differences show. What could be the explanation of these differences, because there are two measures that allow you to explain the phenomenon.
Okay. So first of all, I think for behavioral study, let me start with my behavioral study. Actually, it's quite interesting that we even could find the similarity effect during my behavioral study because, as you know, I use the block design and I mixed up all the similar pairs and dissonant pairs. But even on that case, I still found quite robust similarity effect. But during the EEG measurement, as you has already pointed out, the similar effect seems only revealed at some certain electrodes and time windows, and not as learning effects across maybe even all the S2, S3 window, and it only occurs on, for example, temporal parietal clusters. So I think in my EEG study, which shows that for the electro clusters I have picked, they are actually quite, this brain area, they actually have different function. For example, for some electrodes, they are more likely to deal with the audio-visual integration, and for some certain electrodes, they are more likely to process the characteristics of the sounds. So that's why maybe we only find in some certain time window and some certain electrode, we will find the robust similarity effect. Yeah, that's
Do you could you entertain the possibility that the behavioral results may simply be a lot later in time than the ERP results, so that the brain responds before we act?
yeah, that's a very interesting opinion. Yes, I think that might be yes. Yeah.
And what would that tell us?
So actually, I think for our brain, for example, in the early time window, we have already processed the characteristics of the stimuli, which means we will our brain will understand the philosophy. Is that word? The characters of the stimuli, and they will understand the differences, even the distinct differences between the stimuli. But for our reaction that might be not so sensitive as our brain did. Yeah. So actually, I agree with your opinion. I like this idea.
Yeah.
Yeah.
It's always a very interesting result when the brain does something different than the button press, right? All right. You did give one explanation that I would like to follow up a little bit on, and that has to do with the rationale of choosing your specific combinations for analysis. So you used a combination of curve fitting and ANOVA-based ERP analysis. And I wondered, also now that you're saying, it only showed at some electrodes and in particular time windows, how do you evaluate this approach after you're done it? Would you think this was the optimal choice of analysis, or would you think about it differently now?
Highly esteemed opinions. Thank you so much for your question. So if I understand correct, you asked me about the curve fitting method and whether I still think this is a good way to evaluate.
All right.
Yeah. Actually, for me, I still think for this kind of accuracy curve, it would be a good choice. I use a power fit in my case, but actually I also tried different kind of functions. And this kind of function actually perfectly matches this kind of learning curve. But if we change to another kind of task, it might not so perfect. We might have some different choices better than this.
Okay.
So I think it depends on different tasks. Yeah.
But you don't think it overfitted the behavioral data?
According to the parameters, it shows quite good fitting results. But yeah, maybe as you said, maybe for accuracy, it would be a good choice. But for response time, maybe it's not so good. Yeah, I think I agree with you that for accuracy fitting, it might be a good choice, but for response fitting, maybe, not so good. Yeah.
And in terms of the choice of your time windows for the EEG analysis, do you think those were good time windows? I mean, what motivated you to choose these particular time windows?
Yeah.
Did you look individually, and did you choose an average across your individuals? Or, sometimes people have different latencies, they have different onsets. So how did you potentially deal with that variability?
Yeah, thank you so much for your question. I actually, in my study, I used the GFP method, which is ground field power method.
Okay.
Across all the electrodes, all the participants, I use this method to see at which time window will have a most strong effect. Yeah, and I also provided a plot in my thesis, and we could see that from S1, S2, S3, they show more strong brain activation. So that's why I chose this 3 time window.
Okay.
Yeah.
It's a very good answer. Thank you very much for your for this discussion and your nice answers. Thank you.
Thank you so much, madam.
Thank you, Professor Kott. I pass on the word now to Professor Dr. Jarmo Hämäläinen, expert in cognitive neuroscience at the University of Jyväskylä in Finland. Jarmo.
Yes, thank you, and greetings from Finland.
Hi.
I was also very happy to read your thesis and found it very interesting. And my question is a little bit more theoretical, and I was happy to see that you had described different types of theoretical frameworks in your thesis. So you mentioned there, for example, in addition, of course, to this multimodal integration, you mentioned also the phonological theory and the multi-deficit model in dyslexia. So I was then just wondering or thinking, like, how would you place your studies on this multimodal integration in relation to this multi-deficit model of dyslexia that kind of incorporates different types of areas of cognition, and then this multimodal integration would be one of them. So how do you see that these fit somehow together?
Okay. Highly esteemed opponent, thank you so much for your interesting question. At first, I would like to repeat because the connection is not so perfect. So you said here is a theoretical question, so you might ask me through my study which, for example, which theory I would support, or which theory I would challenge, or is that?
Yeah, or I was thinking, like, because the current theory is kind of emphasizing this multi-deficit model. So how do you see now your studies in relation to this multi-deficit model?
Okay, so yeah, I still not really sure I understand correctly, but
I think it's he mentioned, how do you see it in relation to a multi-deficit model? Yeah, that was a bit unclear in the audio right now. Yeah.
Yeah. Thank you so much again, Professor. Yeah, actually, through the results of my own study, I would say that I quite like multisensory model because according to my study, it seems that it challenging the core deficit model that phonological processing is a core deficit of readers with dyslexia. And if according to that theory, in my study, readers with dyslexia and typical readers, they all show quite robust similarity effect during behavioral task and also the brain activation. So yes, so for my part, I think from the results of my study, it did challenges the core deficits theory of phonological is the most important thing in dyslexic study. I would like to say that actually for me, dyslexia is quite a complex disorder. So it might be the mixture of many aspects. So for example, in my study, I only include participants from most of them are from university. They are highly educational participants. But if I could have a large sample of participants, I would like to find if they have more deficits on, for example, on audio or visual processing. They might show differences when they did the same audiovisual learning task. So back to your question, I think for me, I quite like multisensory theory, and also I think my results partly support this theory. Yeah.
Yeah, thank you. Indeed, it would be, as you mentioned, really interesting to see then if individual would have more deficits in the phonological domain or working memory domain, like how would they then handle this multisensory integration? I think I have time for one more question.
Yes, you
you
do. Yes, please, go ahead.
Yeah. So this is related to the learning task that you used, and considering also that your participants were adults. So I was just wondering, how do you see then, like, could this learning process be different if you would have studied children? Like, how do you see that this would link then to the actual reading acquisition process that kids go through?
Highly esteemed opponent, thank you so much for your interesting question. Well, actually, that's what my colleagues are currently doing. They are using quite similar task to do a longitudinal study, and they tested the children from a very young age. And as you said, actually, in my study, I tested adults, and they have maybe already have some compensatory strategy, which made them could complete this task successfully. But in my colleague's study, even they are still doing this study, but the preliminary results is that some children would show some challenges during the learning phase. That's to say, some children may be at the beginning, they will find this task quite challenging, not like my adult participant did. So for me, I think children with family risk of dyslexia, they might show both at the learning level, the initial learning level, and at the post-test, the automatization phase, they might all show the challenging compared to their peers with the same age. Yeah, that's my hypothesis.
Yeah, thank you very much.
Thank you.
Thanks a lot, Professor Hämmerlinen, for this nice set of questions. Also, your time in preparing all this and reading, highly appreciated. I pass on now the word to Dr. Lars Hausfeld, expert in auditory neuroscience of this university. Lars.
Dear candidate, congratulations, first of all, to your work. Congratulations also to your supervisory team. It was great to read a very nice series of studies. You're doing a very thorough EEG study, really at the core of the thesis, I feel. You have then a proper second follow-up study, let's say, on this one on the behavioral level, and you finish off with a very impressive data set and analysis of the data set for fMRI and recalibration. So congratulations for that. For my first question, I would like to ask one of the participants to come and read out proposition 7.
Cross-linguistic validation is essential for establishing universal versus language-specific mechanisms in reading research.
Okay. Yeah, thank you very much. This proposition you put up as also for the field, I guess, in dyslexia research. And I would like to ask a bit, what do you mean by cross-linguistic validation, and why is it essential for establishing this universal versus language-specific mechanisms in reading research? So, what was behind the proposition?
Esteemed opponent, thank you so much for your interesting questions. Yeah, and thank you so much for your kind compliment. Yeah, I really like your question. Yeah, as you all know, actually, Dutch is quite a transparent language. So for me, I'm really curious that if we did a similar audiovisual learning task using different kind of language, whether that would affect the results. For example, because I'm Chinese, I will use Chinese as an example. In alphabet language, we only have 26 letters, and that's enough. But in China, I think we all know thousands of characters. They all look different. And what is more challenging is that in some Chinese characters, they will have a part of character inside with the similar sound, but another half of Chinese characters, they might have also the same part as the last one, but they pronounce differently. So my hypothesis that in different kinds of language, for example, in Chinese, maybe people will more rely on the visual information because they just remember everything. And in some language with quite a transparent orthographic, such as Dutch, maybe people will more rely on the auditory information. So maybe phonological similarity at this point may play different role in different kind of languages. For example, in Chinese, maybe visual similarity would be more important. So that's why I think it's important to use to investigate the same question on the different kind of background, language background, and to say what will happen.
Yeah, thank you very much. Yeah, these are some interesting ideas, I think. So I was then wondering, so if these writing and reading systems in different languages might be very different, right? You mentioned Chinese as an example, then you have Dutch as very transparent, you have French as very opaque, you have some which are syllable-based and so on. I was wondering, so how does dyslexia then show up in these different writing and reading systems? Is it all very similar to the Dutch system? So I'm naive to the field, right? So that's why I'm asking a very naive question. So how does dyslexia look like then in these different systems? And does it have maybe consequences also for diagnostics? Or is it still all based on the very same neurological processes, which might be atypical?
Esteemed opponent, thank you so much for your question. Let me provide a little bit background. You know, in Chinese, we have two kinds of dyslexia. We have Chinese dyslexia and English dyslexia. So actually, they are kind of overlap, but not the same group of people. So which means actually language is a quite complex process. So maybe some people, as I said before, they are quite challenging to handle visual information, and for them they will feel confusing. Look at the Chinese character which look like so similar. So another kind of readers with dyslexia in China, they're suffering from the English dyslexia. So I think in that case that would be really challenging to process English and to distinguish between each sound. So phonological processing might be really important in that case. And also, as my study did, to integrate visual information and auditory information to automatization and consolidation, this kind of established pairs would also be a great problem for this kind of readers with dyslexia.
Yeah, thank you very much. So I think when I was not thinking a bit about it, I think it also fits interestingly into this multi-deficit model, right? Because I guess different writing and reading systems impose different resources to be used for reading and writing successfully. So it would be interesting to see what kind of different demands different systems basically place on the reader. And then how this maybe is then manifested in the dyslexia. I think I have time for one more question. So I would then, what do I do? Let's go to chapter 2. the EEG chapter. And coming from auditory neuroscience myself, I was a bit puzzled, I have to say, by the choice of your channels of interest that you had. So these are all very much lateral clusters. And for example, when I'm usually doing EEG and look at auditory responses, I always use a frontocentral cluster first to establish, okay, we get an auditory response. And also looking, for example, at audiovisual integration, also with theories that auditory cortex, especially also there might be early effects which reach out into auditory cortex. I was wondering about the choice and maybe why one was not looking at this very, let's say, established, I would say, auditory cortex cluster.
Yeah. Esteemed opponents, thank you so much for your question. In my case, I chose this electoral cluster based on the previous literatures of our field. Actually, it's from my very respectful members of committee, Professor Kalipidi and Professor Hammerleis. Yeah. So according to their previous literatures, I could find that frontotemporal, temporal parietal and occipital parietal, these areas is really important for audiovisual integration. So why I also would like to investigate these clusters, and also I found quite interesting effects across these clusters. So I think my choice is correct.
Yeah. No, for sure. I mean, it's for sure one way to go. But for example, what you were doing, what you were also already nicely describing to the question and as an answer to Professor Kotz, was that you are using the global field power to define your time points of interest, so to say, right? So it would be interesting to look just at a single time point and see how does the topography and not only the clusters look like. And let's say in a good case, you basically show, okay, it's a good choice, you know, that's where the interesting action starts. Or you actually see maybe that there are also other regions or other channels which might be interesting. Okay. Then thank you very much. And congratulations again.
Thank you very much, Dr. Hausfeld. I pass on the word now to Dr. Iliana Karapidis, expert in developmental cognitive neuroscience from the University of Zurich in Switzerland. Iliana, you have the word now.
Yes, hello, good afternoon from Switzerland. It was a pleasure to read your thesis. Congratulations on the interesting work you've done during your PhD. I would like to start with also a follow-up question on chapter 2, which you already have discussed in previous questions. So you report no significant difference in the way that adults with and without dyslexia learn audiovisual associations. But individuals with dyslexia show difficulties when applying these learned audiovisual associations in reading tasks. And you argue in your thesis that this might reflect not a pure deficit in learning, but a deficit in retrieving the learned information. So my question is, how could we experimentally test this proposition of a deficit in the retrieval process? Which experimental paradigm would be suitable, and what kind of design would you use to do that?
Esteemed opponent, thank you so much for your compliment and for your interesting question. First of all, I would like to repeat your question. So you asked me about, in my study, in my case, I found that the differences between groups not occur on the learning phase, but shows for the outcome measurement, when they are applying these newly learned associations, I found some difficulties. And you asked me whether it is only because of different task design. Maybe if I choose another kind of task, that will also affect the observation. Is that correct to understand?
Yes, and if you think about how you could take this a step further to study exactly this step of applying the knowledge, applying what you've learned. So one of your ideas was to explain this by retrieving the information. So how could we study this aspect of learning, not the learning process per se, but how the learned information is then applied? How would you do that?
Oh, so it means such as for early prediction or intervention to use?
Or a new experiment that you can use? Can you think of?
Yeah, so only for my experiment. Well, that would be a very interesting question, actually. Yeah, to think about that, to how to expand my study. Yes. So first of all, I have to say that in my study, in my case, I only let me provide a little bit of background. I only ask a participant to read for 1 minute. It's not a very long time, but still readers with dyslexia show some challenges. So I was wondering maybe that would not be a good idea to use some tasks which are really long. And so if I will design another kind of application task, I still would keep it as quick as I can, just like a flashcard. So for example, to show different kind of flashcard and to ask the participant to respond really quick to this quick stimuli. So I don't think I will use a super long post-test to test it. I think speed is quite important. Quite important aspects in this kind of study, and then because in my study, I use half of the matching pairs and another half of non-matching pairs, so that would be 50-50. So I think that would be interesting that if I could change the portion of this matching pairs, for example, from very less portion to a lot of mismatching pairs, and to see whether that would affect the learning. And during the post-test, I would show both the matching pairs and non-matching pairs and ask the participants to choose. And also there are some kind of task design to show the two symbols on the screen at the same time and ask the participant to choose one of them. When I design my study, I also consider about this kind of task, but for EEG study, it's not so suitable, but for behavioral study, to choose the correct symbols within a lot of symbols, I think that would be a quite interesting post-test. Yeah, thanks.
So you would do that purely behaviorally, or would you mentioned that EEG might not be the ideal method of choice. What about perhaps using a different imaging method? Do you think it would be interesting to look into brain processes during that experiment?
Yeah, I mean, for EEG study, that would be not a good choice to present two symbols at the same time, because that would be confound. Yeah, that's why I mentioned about that. Yeah.
And can you think about another methodology where it might be less problematic?
Sorry, you mean?
What about another imaging method? Do you think you would have the same challenge with MRI?
Yeah, MRI would suitable than EEG in case I would like to use this kind of design. I agree with that. Yeah.
Good. Then I would like to move on to a second question. The experimental paradigms that you used in your thesis are all audiovisual. And my question is, are your findings specific to audiovisual processing? Or can we also maybe draw some conclusions about multisensory learning processes in general? What if we use a similar experiment or similar experiments with people who are blind, and instead of visual information, we would provide them with tactile information? Would you expect similar effects?
Esteemed opponent, we're still talking about chapter 2, right? So if we...
Yes, either the artificial symbol speech, or you can also think about a version of the recalibration task. I think it can apply to both, but we can focus on the learning task, yes.
Yes, because I think for my study, it more involved different kind of visual cues. And for my EEG study, I control the visual similarity between the stimuli, but if I use visual similarity as a factor, I think that, yeah, indeed that will also be interesting. And I think disparate visual input or visual information, that will be also interesting to add some, for example, action, for example, to draw the letters during learning. Actually, when I did a research practical with Francisco, my supervisor, our group of students, we use this kind of task that we will present the audiovisual pairs, and at the same time we will present the video that someone to use their body to draw the letters. And also from this kind of study, we receive very interesting results that if the learning phase could add some more multisensory context or multisensory cues, that participants would learn about the audiovisual pairs more efficiently. So for me, I'm a big fan of multisensory theory. Yeah.
And in case of individuals who are blind, you would not be able to use audiovisual, right? You would have to use audio-tactile. Do you think that the brain processes that you studied and the learning would look similar, or what would you expect?
Yeah, actually, in Helen Heller's book, she learned language by touching her teacher's tongue. So as you said, when we cannot provide enough visual input, we could use other kind of cues to help learners to learn. So I think, well, if we would like to support the readers, we could find all kinds of additional information or additional cues to add a more rich environment for them to learn. Yeah, just like Helen Keller's teacher. Yeah.
And what happens to the brain?
Yes? I get your point, but we have to move on, because otherwise colleague Schumann would not have a chance to address her questions. Is this okay? Sorry for cutting you off. Thanks a lot to Zurich. I pass on now the word to Professor Schumann, expert in clinical application of non-invasive brain stimulation, also from this university.
Thank you very much, dear candidate. I also want to congratulate you for a very nice thesis. It was very interesting to read, and I especially liked how you framed the overarching question across chapters. I also want to extend my congratulations to your supervisory team, Professor Bonte and Dr. Gentile. Very nicely done. I have a methodological question about the construction of your similarity manipulation, because this is something you use a lot throughout this thesis. And if I read it correctly, you define phonological similarity based on acoustic resemblance. And you actually do this using an independent auditory similarity rating approach, right? So people rate it from 1 to 5. And the first question I have is, why do you use this approach rather than a phoneme discrimination task?
Highly esteemed opponent, thank you so much for your kind compliment, and thank you so much for your interesting question. Yes, I agree that the discrimination task would be also a good choice. And also when we design this task, we have considered about whether to use this kind of task as our pre-task. And maybe I could first explain why our final choice would be this kind of pre-task. Mm-hmm. That's why, because at first we have 22 different syllables. If we use, like you said, discrimination task, that would be quite a lot combination, because we need to make a pairs compression and to overlap all the combinations. That would be a lot of trials. And for now, we have 22 syllables and we have 330 trials. During the task, participants are already quite annoyed. So I think I agree that discrimination tasks would provide a more reliable information. But if when we do the pre-test, we would like to really define which groups of syllables are perfect for a similar group, which are good for different groups. So we choose a very large pool with 22 syllables. Actually, it contains some redundant information. So we use this kind of way to make sure we could make the good choice. That's why we use that 5-point scale.
Okay, that's very clear, thank you. And if I read correctly, you did this with the typical reader, right? Not with the dyslexic reader. And I wonder whether this would have made a difference. So would you or should you also have done this with a dyslexic reader? Because the similarity structure itself could maybe already differ between groups. What do you think about that?
Highly esteemed opponent, thank you so much for your question. Yes, indeed, I think your consideration is really reasonable. And actually, because of the same consideration, we set a dictation task and a conversion task, just like pre-test for both EG study and for behavior study. So after the study, we will ask 2 groups of participants to do the similar task, and we will record their judgment scoring, and we will compare their dissimilar groups scoring with the pre-test, and actually it replicated the findings during the pre-test. And another consideration is that we are using the familiar Dutch sounds as our auditory stimuli, which means for all the participants, they are quite clear stimuli and they are easy to understand. So I think your consideration is really good, and also maybe because we are using familiar Dutch sound, so that would be acceptable. But if we are using unfamiliar sounds, I think that would be very important to make sure our pre-test participants include Dutch participants.
Mm-hmm. Okay, thank you. Then I have a question about chapter 4. And also, I am not an expert at all in dyslexia, so maybe what I say is completely stupid. But I'd like to hear your thoughts about this. So in your study, you actually report increased fusiform activation in dyslexic readers during the lip reading exposure condition. Now, if I think about dyslexia, I think, well, there could especially be a mismatch between what I hear and what I read, but they don't really have any difficulty in understanding spoken language. So I was very surprised to actually see a difference there between the 2 groups. And I wonder how one could interpret this effect in the fusiform area. Does this tell us something about the underlying mechanism? What is happening there?
Highly esteemed opponent, thank you so much for your interesting question. And yes, I would like to share my perspective according to these findings. Well, actually, in lip movement condition, we find that there are quite significant differences between groups, but in text condition, we haven't found that.
You can shortly finish your answer if you like.
So actually, it's a kind of a compensation for dyslexic reader, which means they are not so efficiently handle this kind of audiovisual integration task. That's why they show this kind of pattern. Yeah.
Okay, thank you very much.
Thank you, Professor Schumann. Dear candidate, the time appointed for defending your thesis has passed. The degree committee will now withdraw and discuss the quality of your thesis and your defense. And I kindly ask you to await our return here in the audience together with your friends, colleagues, and family. Thanks a lot.
Thanks for asking me.
Yuewei Cao, the degree committee here present, has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Bonte is now authorized to confer upon you this academic distinction in accordance with the Dutch university custom. I kindly ask your supervisor now to take the floor.
All right, please. So I first say a sentence to which you give an answer. Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Yuewei Cao, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary and the other members of the committee and affixed with the official seal of the university. Yes. Dear Yuewei, Dr. Cao, on behalf also of Francesco, congratulations with this great achievement. And now I hope I'm saying this correctly. Gongji, gongji. To you, your mother, your family, and your partner, Han, who I think is present online. So, how did this all start? I went back and found an email of Manli in October 2019 saying, there is a girl, Yuewei Cao, CV attached, from my previous lab who is interested in applying for a CSC grant within our group. So we organized an online meeting to discuss possible projects on audiovisual learning and dyslexia. Already back then, you listened in your typical Yuewei manner. First a bit shy, hesitant, waiting everybody's words, and then making up your mind. After your graduation in the summer of 2020, you first started your project remotely. Then, after the world opened up again, after the COVID pandemic, you made the big step to move all the way to Maastricht. You immediately conquered our hearts with your kind smile. But at the same time, you seemed very good at ignoring some of our advices. Later on, you admitted that indeed at first you silently doubted our approach. As you once described it yourself when you started working in our team, your perspective was fully data-centric. But then, and here I again quote your words, you began to perceive the research journey as a harmonious dance between data and human connection, which is a great example of your beautiful way to express yourself and of how you opened up and became one of our team. You took your PhD journey as a real challenge, step by step, overcoming every hurdle. You learned how to work with behavioral, EEG, and fMRI data, which I think is really admirable. And for example, for your behavioral study, you tested dyslexic participants not only in Maastricht, but you also went to Utrecht, Den Haag, and Amsterdam to collect your data. And you somehow also managed to convince the founders of the Dutch Hoy Foundation for Dyslexia, Tamara and Stefanie, to travel all the way from Amsterdam to Maastricht to participate in your EEG study. And Tamara even took her daughter as an extra participant. Other participants came all the way from places like Venwalden in the north of Friesland, traveling a total of 676 kilometers, I have this of the participant form, to participate in your study. Right? So you must be very convincing. So the writing was the last hurdle to take. And I think I'm not exaggerating if more than half of it still needed to be done when you moved back to China April last year, right? And from May onwards, I was then also positively surprised by the continuous flow of updated chapter versions in my inbox. Right? It was really continuous, all the new chapters. And they were always accompanied by beautiful, polite, very thankful, but also quite directive emails. And you actually managed to finish your thesis by the end of July, which is really an amazing achievement. And I'm very proud of that. Dear Yuewei, you are a strong and unique person, with definitely many more sides than people realize when they see your sweet schoolgirl appearance. One special moment I still remember, for example, is encountering you at the Oxfordlaan 55, dressed in this beautiful black dress, with matching makeup and a hairstyle. And your originality does not only show in the way you dress, but also in your way of expressing yourself, both non-verbally and verbally. It is also visible in the way you approached traveling in Europe while you were here, with Italy as one of your favorite destinations, and I can also relate to that. You even took your mother to San Siro in Milan, and I can relate a bit less to that, but I find it anyway very, very cute that you went to the AC Milan football match together. And it's not the Milan part, but it's the AC Milan part. So you watched this game together with your mother, which I really find really nice that you did that, and admirable. We miss you in the language group. We miss your giggling at social events and your big smile when ordering at least 2 cookies for breakfast or lunch at Bandito, especially a big smile when Quinten or Sean were there. And I will always cherish your, "Can I give you a hug?" after some of our more personal conversations. On behalf of the whole team, I wish you all the best of luck and happiness for your new life back in China. We are very curious to know about your upcoming new job adventures at TikTok. Congratulations again, be happy, be very proud of yourself, and don't forget to celebrate.
Thank you.
Yeah, also on behalf of the Faculty of Psychology and Neuroscience and Maastricht University, congratulations to this wonderful achievement. Now take a deep breath and just enjoy the moment. I would like to thank the members of the reading committee, all the way stretching from Finland to Switzerland, the supervisors, even coming home, if I may say, from Rome, the colleagues in the audience, people from Banditos are there, the mother is here. So I think it was a very nice event. I formally now close this academic session. Wish you all the best. And a formal remark is that we don't have the towards the stairs or already go towards the cafe. That's up to you. But we take first some pictures so that this moment also gets frozen into the digital domain so that you can upload it at TikTok. Huh?